In this short tutorial, I want to show you how to identify the dataset and then find the data that you can download and prepare the sample metadata for the analysis. Okay, so what I'm going to do is I'm going to type in here the name of the publication, the date, and the author. Modeling Precision Medicine, Damon, 2013. So what you can see is I'm finding the actual paper. So I'm going to click on here. This is under PubMed. And then here on the side, I can find the geodatasets or the SRA. So let's go over here. It will give me a detailed view of the datasets. And I'm going to be looking for expression profiling by high throughput sequencing, but this says other. So in fact, what I'm going to do is I'm going to take this one, expression profiling by high throughput sequencing. And we also know that it's 56 samples. So let's click over here. Now this is going to be the data set. You can see the descriptions of the cell lines, that this is RNA-seq data, and these are all of the samples. Let's go over here. Bioproject, that's where I want to get to, PRJNA 210428. So I'm going to click through over here. Okay, and now I can click on the 56 SRA experiments, and over here I will go under Send to, File, and here I will do Run Info. Create file. So this is what's in the file itself. You can see all the download path right here. It will provide me links to the SRR data for each one of the samples, and it will also give me the other information that I need. For example, that this is pair and reads, and the names of the samples and what organism it should be mapped to. The files that we have are already uploaded to the system. So now we are going to take the SVL file that we have, and we're going to create our first RNA-seq pipeline. Let's go over here under RNA-seq/chip data, and we're going to make the selections according to our dataset. So first of all, as you remember, this is pair and reads, and we will be using the model genome being human genome, so we will use the latest assembly of the human genome, GRCH38, and we're going to upload files and groups. And because it is already uploaded to the system, we're actually going to use this URL bulk upload. So let's take our SVL. We're going to have cloud and low versus normal like samples here. Define groups check. We actually don't need groups because we're not doing differential gene expression. And now let's create our pipeline. So the first option, now pay attention, every time you click there's important information right here that tells you a lot of information that you can learn from about the process that we are going to be going through. Now the first thing, as we saw in the lecture, is to prepare the trim unnecessary amplification. So we want to first of all use Trim-O-Matic to clean technical sequences. And these are sequences that are stored in a databases. And again, you can click on this link and read about Trim-O-Matic. Let's click on Save. The next thing that we want to do is we want to reduce the duplicate reads from raw sequencing data. How do we know that these are technical duplicates or replicates and not actual mapped reads on the sequence? Well, because they all align perfectly in one space. And when they're perfectly aligned, that means that this is something technical, because normally reads are kind of overlapping, and parts mapping here, parts mapping there, and they're different lengths, etc. So this is how we know that this is amplification. We want to get rid of that. Now afterwards, we have several options. We can either map on genome, and then we would have to detect the exons and map on junctions, and then construct the isoforms. Or we can simplify the problem and focus just on mapping on transcripts. And transcripts are already recorded in the GTF file, like we saw before, and we're just going to see whether the reads map to those places or not. And if not, we'll just leave them as non-mapped reads. So what we'll click is bow tie to T. So you can see that bow tie to T is again explained here, how exactly it maps, and you can go and read about the algorithm itself right here. So let's click on Save. And the final thing that we need to do after we map those reads is to quantify the transcript abundances. And so you can see here that the previous algorithm created the SAM file. So this is the sequence alignment that was produced there. And now we are going to quantify how many reads fall onto each one of the known sequences. Now, quantification is not as straightforward as you think. Quantification of reads requires us to have some expectation of general noise. So we expect the reads to be placed in many different locations at approximately the same rate. But if there's an abundance, if there's some kind of a natural variation that is actually in the data, we would expect that to have some abundance above that level of noise, and that is what's called by expectation maximization. The other thing to remember here is what values do we want to use when we do this quantification? Do we want to just have expected count, or do we want to use some normalization? So normalization that we have optional here is fragments per kilobase per million reads, or TPM, which is transcript per million. So we're going to use FPKM, click on Save. You also can create an exon table, but you don't have to, and click on End. And now we have our first pipeline, so let's call it RNA-seq first pipeline. Click on Save, and now we can click on Run Pipeline. So now this pipeline is running. It's important to note right here, 1% started, and also if I go under my pipelines, I have to see that it is green, and it is indeed processing. So this is how you create the pipeline for RNA-seq. Now there are several other options as well. As I mentioned, we can map on genome, or even we can use alternative ways to quantify. So if you go under RNA-seq 2, you will notice that we added here also sailfish and HT-seq, which are alternative methods of quantification.